This is our first online recording for pathophysiology. The first chapter has to do with cellular injury. So just to acquaint us with this topic, I'd like to do a brief overview of the cellular organelles. Some of us may have not thought about them for a while, so we are going to go ahead and do a little bit of a review into these organelles. Whenever you see a blue link, these are live links that you can click on. It takes you to a page or to a movie in some cases, but I'm going to give a brief overview of each of the important parts, which if you take a look at this slide, part one of this lecture involves the overview of the normal cell. The lecture that we'll do in class will have to do with the pathophysiology of cellular injury, which has to do with damage in some cases to these organelles or all of the organelles. So I'd like you to be able to have some familiarity with the cell components that have been discussed. Importantly, I want you to know what the primary function of those are. So in pathology, you look at whether or not those things appear normal with sight, if they're in the correct amount, the correct size. So there's a lot that will go into actually looking at microscopy of cells and tissues. So if we start off, we're just looking at a eukaryotic cell on slide 3, and there's eukaryotic and prokaryotic cells. You can remember that you, as a human, have eukaryotic cells, and eukaryotic cells are made up of some different components, a plasma membrane, we have the nucleus of the cell has the ability to replicate, it contains genetic material in the nuclear areas and also in mitochondrial areas, and then it's also responsible for manufacturing new cells, and the major building blocks of cells and pretty much all tissues in the body are proteins, and those things are going to be created by ribosomes, which are associated with the endoplasmic reticulum and are stored in the Golgi apparatus. So let's go through those very quickly. Starting on the outside with, as a pharmacologist, my favorite part of a cell, the plasma membrane. So the plasma membrane is a semi-permeable or selectively permeable barrier, meaning that some things can get in and some things cannot. And that's important because we'll talk in this chapter or later chapters about some cells being excitable cells, and some cells just being, on all the time. Like a liver cell can always be a liver cell. It can always do liver things. However, a muscle cell, we turn those on. They're excitable. We don't want muscles to be in a constant state of contraction that would cause some spasticity or an increased tone. So that turning on and off of cells has to do with the charge of that cell, which is determined by the concentration of ions on the inside versus the outside of the cell. The plasma membrane is a very highly lipophilic structure, meaning that fat and fatty substances can diffuse through it very easily. That's because it's 97% phospholipids. And if you take a look at this picture over here, you see those little things that look like dots and then these two legs underneath. Those are phospholipids. And what you have, remember, your body is water, so there's water everywhere in the body. So we call these heads, they're hydrophilic, they're water-loving heads, whereas these tails are hydrophobic, so they don't like water. And there's some fun comics that I'll show you once we get into class where you can remember this because if you picture a water gun fight inside of the cell membrane, they will contract their legs because they're afraid of water. So if you just drop a phospholipid into a watery area, they orient in this way with the heads facing outside towards the water and the tails facing inside where they are protected from water. The other 3% of what makes up this plasma membrane, besides the phospholipid components, is cholesterol, which is very fatty. So things that are fat soluble, meaning that they dissolve in fat, they pass very easily through this barrier. And they do so generally through just simple diffusion, which means they go high to low concentration. And just a reminder for you that there are some things that you should remember about the cell and what's high inside versus outside. And if you remember it, that will help you quite a bit in your life. But potassium is really, really high inside of cells, and sodium and chloride are very high outside of cells. So that means sodium and potassium would have the tendency to move in very easily, and potassium would have the ability to move out down its concentration gradient very easily. And in order for the cell to say the correct size and the correct charge, we need to control that free movement of ions. So these are very tight controls. So some things can engage in diffusion, other things have to take these specialized transporters. So there's different channels embedded in the plasma membrane, just large proteins that sometimes span the barrier, and they're selectively gated to let certain things in and out. And the general term for things going inside of the cell, like you can see in this picture down at the bottom, is endocytosis. We break down words a lot in this class. Osis means the development of. Cyto means cell, and endo means within. So developing inside of the cell, endocytosis. When we take a look at the cell membrane, bringing something of large structure into the cell or of liquid structure, the actual membrane forms what we call an invagination. So it pockets down, and this is all our phospholipids here. And essentially, what you will see is there's also some actin and myosin embedded in here. And if you've heard those terms before, actin and myosin means that it has the ability to contract. So once we fill up this invagination with whatever substance is being brought into the cell, this vesicle will snap shut so that we now have the substance contained within inside this structure that it's made out of, the cell membrane. And that's called endocytosis, and the structure is called a vesicle generally. So a vesicle is just a piece of the plasma membrane containing something inside of it. If the body wants to get rid of something, this could be a hormone, for example, a neurotransmitter, it could be debris left over from digestion of things brought into the cell. This same vesicle is attracted to the plasma membrane by certain proteins that are expressed on the wall. And essentially what it does is merge with the existing plasma membrane. And you can actually see the shape here. It forms something called an omega structure because it forms this letter omega. And now we are dumping out the contents into the extracellular environment, and this vesicle, which was once forming a little pocket for this matter or particulate, can now merge back into the membrane. So it just is continuous. We can pinch off a little piece and then we can add it back in later. So bringing something outside of the cell is exocytosis, again, the development of cell external. So bringing things out. So endocytosis is the general process of moving things in by forming a vesicle which contains it. When this cell is an actively phagocytic cell, so something with phage means to eat. So phagocytosis is the development of cell eating. So if this was a cell, maybe it's a white blood cell, a special kind of neutrophil, it would be responsible for phagocytizing things in the body that we didn't want there. So it kind of cleans up the debris, and it would do the same process. It would bring it in, it would form a vesicle with it, and then we'll talk about how it deals with it afterwards. So that involves bringing in something that needs to be neutralized, phagocytosis. Pinocytosis is describing the same process of endocytosis, but it has to do with liquid or fluid. The main example for this is like fat, for example. We mentioned that the plasma membrane is made out of cholesterol. So the cholesterol in our diets, when they're turned into LDL, that LDL binds to receptors that would be inside of this little invagination. And then when it snaps shut, that cholesterol is then brought into the cell. And the vesicle in that case would just contain LDL, which will then be used to build up a stronger, bigger cell membrane. And then exocytosis, as we mentioned, is moving something out of a cell. Okay, so I mentioned that we have diffusion and things go down their concentration gradient from high to low concentration. So remember, sodium is really high out here. So if there was any channel that opened that sodium went through, it would just naturally flow through there. So that's called passive transport. It just passively goes down its concentration gradient. It's very easy. It's kind of like if somebody was cooking a steak outside, the concentration of the steak smell particles would be really high outside, and then eventually they would start to waft into the room that you were sitting in, because they're going from high to low concentration. That's how gases diffuse, that's how ions diffuse, that's how everything diffuses. So because that requires no extra energy, that's called passive transport. So in the case of diffusion, it's just the kinetic energy that's created by the movement of particles. There's also cases, like in this picture, we just have a channel. That's still, it's passive transport. There's no energy that's required for something to move through that channel. However, when things are moving against their concentration gradient, so we'll talk many times this semester about the sodium-potassium pump. It is the most prevalent pump in the entire body, and it is in every cell working all of the time to keep sodium and potassium in the correct concentration so the cell stays the right charge and the right size, and it can function normally. So in that case, the way that the sodium-potassium pump runs is to bring things not down their concentration gradient, so from high to low concentration, it moves them in the opposite direction. So if we wanted to move sodium from inside of the cell to outside of the cell, that wouldn't happen naturally because things don't want to move that way. That's not normal. So what you have is some sort of energy source, and we know that the main energy source in cells is ATP, adenosine triphosphate. And ATP runs these pumps kind of like a Ferris wheel, just running all of the time, so that things can move in a way that normally wouldn't occur given their concentration gradients. And that's important because things are trafficked together. We'll talk about the role in that with the kidneys, with insulin. It's very important. So just remember, active means that there's energy involved. It usually is ATP. It could also be in the form of borrowing the kinetic energy from something else that's going through a channel. So for example, glucose and insulin bind together, and only glucose can go through a channel when insulin is present with it. So glucose engages or kind of borrows the kinetic energy from insulin. So typically, active means that it's against the concentration gradient. Passive means that it's just going high to low concentration. So we mentioned a bunch of these pores. The other thing that you'll see is that there are receptors embedded in the membrane. So here's a nice protein over here. It looks like it has antlers and there's some H's binding to it. When something binds to a site, so in this case, I believe the H stands for hormone, because what else would it mean? Well, I guess it could mean hair or ham. So let's say hormone. So the hormone binds to a site on this protein. Whenever something binds to a site on a protein or a receptor, we call it a ligand or a ligand. You can say it however you choose. As long as you say it with confidence, that's fine. So these receptors, they have an attraction to something based on shape. Everything in the body is based on shape. The closer the shapes fit, so you see how this H hormone guy is a similar shape to this little site. So it has a high affinity, that's the science word for attraction affinity, to this little spot. And when this hormone binds, we have some sort of action occurring inside of the cell. So we have receptors for hormones, we have them for neurotransmitters, we'll talk about them in the immune system, And there's two types of receptors. There's ionic receptors, which are just simple pore channels, and there's G-coupled protein receptors, which do the same basic thing, but they have a G protein associated with them. I'll go more into detail of them later. We have enough to talk about today. So that's the plasma membrane. It's selectively permeable. It helps maintain this nice barrier. You cannot get through it unless you are very small, if you have no charge, and if you can dissolve in fat. All right, moving on, we have the mitochondria, the powerhouse of the cell. They're always orange in pictures or always orange in models. So the color orange should remind you that it has to do with energy, like a fire. And mitochondria is where aerobic metabolism happens, and aerobic means that it's with oxygen. In biochemistry, you learn about the Krebs cycle, which is how we generate oxygen into ATP, adenosine triphosphate, so that it can power the different things that the cell needs to do. The pumps, kinetic movement, the forming of these vesicles, cilia movement, it's all guided by ATP. So really all a cell needs to engage in the making of ATP is oxygen, which means the body needs to be alive and breathing, and the heart needs to be beating so that oxygen is available in that area. And the mitochondria is a dual layered cellular apparatus, essentially. There's an outer membrane and an inner membrane. Crista is a Latin word, and it means shelf, or kind of like a wave. So I want you to think about that, like these crista, they are places that we can store things, and they make these nice little folds. And there's a lot of proteins there that are embedded to help in this formulation of ATP through the Krebs cycle. You'll see when tissues are very active, so let's say somebody started working out and they're running, their muscle cells will get bigger, and they'll also develop more mitochondria because you have more tissue, it's more active, and you'll need more ATP. So in pathology, you look for the presence of mitochondria, the number of mitochondria, and the general appearance of them. You may have heard of mitochondrial DNA. That is a way to track maternal DNA. You don't get information from the genetics from both parents from that, but you do get maternal DNA from the mitochondria. And it's much cheaper than getting your full genetic sample. So you'll see that used quite a bit when people are trying to save money, which you should be familiar with. I know I am. Saving money, I mean, because I'm poor. Okay, going on, we have the granulated endoplasmic reticulum, and granulated means that it has granules. And when a cell is granulated, it just means when you take it and put it underneath a microscope, it has little dots on it. And those dots could be anything. You don't know until you get closer to it. But we do know on the endoplasmic reticulum that those little dots are ribosomes. And ribosomes are the actual things that make proteins. So they sit on the outside of the endoplasmic reticulum. So if you think of the endoplasmic reticulum as a factory, the ribosomes would be the machinery that's actually making the product. And if you remember from your science classes in high school, that is usually the first time we learn about this. Ribosomes are two different pieces that there's like a 30 AG and a 50 AG. It doesn't matter right now, but there's two pieces that they orient themselves together. And like reading ticker tape, or if you've ever played skee ball or any kind of fun game at the arcade, when all those tickets come out, the ribosomes stay static, and this mRNA, which contains recipes for different proteins, just goes spitting out like these tickets. And there's a code that goes along with these, and these three bases together are called codons. And the way that this works is this messenger RNA is essentially a copy of a protein recipe that the body would like to make. So think of this like going to the library, getting a recipe book, deciding you want to make these brownies, so you photocopy the recipe, right? So this messenger RNA is a photocopy of the recipe. And then you want to actually make the recipe. So you take the recipe home and you start getting out the different ingredients. So each codon essentially makes one ingredient. And in this case, 3 different bases, they make one amino acid. And an amino acid is the building blocks for proteins. So you put proteins together and you get these long protein chains we call peptide chains. So you're starting to make a substance. So that could be another cell, that could be a hormone, it could be a whole bunch of things. But you've got your recipe or a copy of your recipe, and then putting that together by reading the recipe and then getting your amino acids. You might know that there's some amino acids we can just have in our body, and then there's some essential amino acids that we have to take in from the diet. So we talk about that more in the nutritional chapters, but for all intents and purposes, the granulated endoplasmic reticulum, which you may have called the rough endoplasmic reticulum in the past, is this factory which houses, for a brief moment in time, these proteins that are created by the ribosomes. So this is where protein manufacturing occurs. And the proteins can be stored inside of the endoplasmic reticulum for a little bit of time, and then they go on to the next piece, which is the Golgi apparatus. But before we move to the Golgi, there's another type of endoplasmic reticulum. It's called sometimes the smooth, but we're big adults, professionals now, so we will call it the agranular endoplasmic reticulum. Whenever you see A before something, that means without. So agranular means without granules. So you put it underneath a microscope and you don't see any bumps on it. So this is different, obviously, in that there's no ribosomes. And in the body, when you see an enhanced amount of smooth endoplasmic reticulum in cells, typically that tells you that the body was trying to get rid of something, some sort of toxin. Maybe that person drank a lot. Maybe they took a lot of medications. The typical number of prescriptions taken in geriatric patients is 7. So you will see an increase of smooth ER. So they break down hormones, toxins, phospholipids. So there's a bunch of different things that they do. There's also specialized endoplasmic reticulums that we'll talk about in different areas. Like, for example, we'll see these in spermatic tissue to help in spermatogenesis. So they do not have ribosomes, so they don't do protein manufacturing, but they do a lot of degradation and detoxification of substances. So as I mentioned, once proteins are created, so if you look down here, we've got the granular or granulated endoplasmic reticulum. So these ribosomes made some proteins. Remember, things don't just float around in the cell. What happens is we make a little bud off of that cell so that it's now contained in a vesicle. And remember, that vesicle is made out of this plasma membrane material, the phospholipids and the cholesterol. So these vesicles, in this picture, it looks like they're just floating. In real life, nothing does that. Your body is very controlled. It's actually walked over on a cytoskeleton. I'll put a link to a video of this. It's really awesome to watch, by a protein named kinesin, which comes from the word kinetic. And once these vesicles get to this Golgi apparatus, just like we saw before, this fuses with the membrane and then flattens itself out to become part of the Golgi. So the Golgi apparatus, this is kind of like the finishing warehouse. So if you think of the granulated ER, this is the factory where these goods were made. They're put in boxes, they're shipped off. This is like your Amazon superstore. So Amazon can put your things in different locations. That's what the cisternae are for, the cisternae. Another Latin word. Latin's very important, and it has to do, again, with shelving. And like a bookshelf, it's somewhere that you put things. And so Amazon can put other things in your boxes too. You don't just have to get one stapler. Maybe you have also your multivitamin being delivered in the same box. So that's what happens here in the cisternae. We can add different things to those proteins. We can add carbohydrates or lipids, or just store it until it's needed. Same thing as a warehouse. Or if the cell has nothing to do, it can just grow bigger. So the Golgi can just butt off empty vesicles to go merge with the plasma membrane of the cell so that cell can grow. So it can house these proteins that were made by the ribosomes on the granulated endoplasmic reticulum. It can house them for a while. It can add things to them. It can store them. And then it will butt off the final vesicles, either containing the products, little additional things added to them, or it can just butt off pieces of plasma membrane to help in cellular growth. In this picture, it's labeled secretory vesicle. Secrete, I like to think of the word squirt. When something is secretory, it usually means that it's being squirted into somewhere like the bloodstream. So a secretory vesicle in this case could house a hormone. So maybe this cell's job was to make insulin, and now it's going to merge with the cell and then be released. Or the same process happens with like thyroid hormone. Very good. So that's the Golgi. Speaking of endocytosis and vesicles, we have the lysosome. Lys means to break apart, and some is kind of like a body. So a lysosome is essentially the stomach of the cell. So lysosomes are very important organelles because it's their job to digest debris. So here's a phagocytic cell. It has brought in some particulate that we need to degrade. In this case, because it was eaten and now it's in a vesicle, this is referred to as a phagosome, which is very nice. That's super descriptive in eating body. And when we bring in this virulent material, like maybe this is gonorrhea, I have a picture of that in real life in the presentation I will show you in class. We don't want that hanging around in the body. So it will be brought into a cell, and this little guy down here that looks like a chocolate chip cookie is not. That's our lysosome. And lysosomes are filled with really strong digestive enzymes. Some are filled with peroxidases. If you know peroxide, you put it on a wound and it starts to bubble. That's because it forms oxygen and can kill things. And some are just hydrolytic, which means it breaks down watery type of substances. So the lysosome, it's like when you eat something, you swallow it, it's brought to your stomach, which is full of acid, and then it's degraded. So your lysosome is full of acid. In this case, we've got the phagosome coming down here, and the lysosome merges with it. So now we have a digestive vacuole. So basically, this lysosome is this stomach of the cell. It's full of really strong acid. It's got a nice strong membrane so it doesn't digest itself, and it will help to degrade substances that are brought in so that there's only debris left over. There is a name for this leftover debris that you can kind of see out here. They're called lipofuscin granules, and a little bit of them is normal. It's kind of like feces, like having some feces outside of your body is normal. That's where it should be. However, if there are little dots, if there's lipofuscin granules inside of the cell, that's bad. Same thing with fecal material inside of the body. And I don't mean inside of the GI tract, I mean like outside in your extracellular fluid. That would be very terrible. And we'll learn about that. So in all cases, having lysosomes, having sufficient lysosomes means that things can be digested and the cell can be kept clean and free of debris. All right, I mentioned the cytoskeleton before. The cytoskeleton is so cool. Actually, this is a link right here to that movie I mentioned, but I will put it up on Canvas so you can access it easily. You have all of these little filaments that form a skeleton of the cell, and they're there for a couple of reasons. So first off, they maintain the structure of the cell. So in the same way we have skeletons that keep us upright, the cells have skeletons. And if we had all of our bones crushed, that would suck, A, but B, we would have no structure or integrity. Same things with cells. So we'll talk a lot in the cancer chapter about pressure atrophy being a way that tumors move and take over spaces, or in crushing injuries. If this skeleton is just destroyed or broken down, then that cell is going to eventually die as well. And you can also see these little black guys look like caterpillars. That's kinesin. And what kinesin does is help to move vesicles from one side of the cell to the other. There's also microtubules in this area, which means very small tubes, and "-ule" at the end of something means like it's really little. So microtubules, those are literally small tubes, and things flow through them, like the T-tubules you learn about in muscle. So cytoskeleton is important for structure and for movement of material through the cells. All right, finally, we have the nucleus, and the nucleus houses, again, besides the mitochondrial DNA, the major genetic material inside of the cell. This is a very, very poorly permeable thing. So not a lot can get into the nucleus, which is good. We don't want things getting in and then messing with the DNA. When things do that, we call them carcinogenic, which you might know means cancer causing. We don't want things messing with our DNA and ruining our recipes for everything in our body. So it's the internal regulator of the cell in that it houses the important material. It's not like the brain. It doesn't tell the cell where to go or what to do, but it contains the important information. So the nucleus has a certain amount inside of the nuclear envelope of cytoplasmic material, and the amount of cytoplasm to the size of the nucleus or the nuclear material, we call it the nucleolus in this picture. That's a really important marker for whether or not that cell is diseased. So it's a long day. The amount of material, liquid versus the chromatic material is important. All right, so 2 layers. We've got a porous outer layer, and then we have a final layer on the inside around the inner nucleus, which helps keep things nice and safe. So nice 2 layers. This is where our DNA is, and the DNA we can see here is packaged into these cool little chromosome type of shapes. So if you go to the final slide, you see what these look like. These are our nice little chromosomes, and the chromosomes orient this way because they are wrapped very tightly around these little ball-like structures called histones. So DNA is wound around these little histones, and then it's supercoils. So if you think about winding up your Christmas holiday lights, if you were wrapping them around, I don't know, I wrap mine around a rolled up newspaper. As I mentioned, I am poor, so that's what I use. So you wrap it really, really tightly, and eventually it starts to wrap on top of each other, and then you have a nice long string of these lights that's kind of supercoiled. That's the way that these histones package the DNA, and then they wind together tightly, and what we have is this nice kind of X shape when everything is oriented and fully packed away. And at the end of these, in this picture they're red, those are called the telomeres. And telomeres are the protective tip of the DNA, because in order for something like a carcinogen to get in and actually mess up the basic structure of the DNA, it has to be unwound from the histones so that you can then access this DNA. And then you have to have a protein come in and unzip the DNA and then mess with these base pairs here. So in pharmacology, there's actually drugs that prevent histone unwinding. That's really cool. One cool note also about telomeres is that they naturally get smaller with age. So if you think of these like an eraser on a pencil, the more you use your pencil and use your eraser, the more worn down it gets. So these, just by existing, the telomeres start to get smaller and smaller. just with age. Fun fact of the day is there's a direct correlation between amount of exercise and telomere length, meaning the more people engage in activity, the longer their telomeres are. So the more protection there is against unwinding and then disruption of the DNA. That's pretty cool, right? It is. You should thank me for that nugget of wisdom. I will accept it in class. All right, we know that DNA is double helical. It's anti-parallel. If you've never learned that, that's okay. You can look on Wikipedia. And then we have these two base pairs that go together, and it's always supposed to be A and T and G and C, adenine and thymine, guanine and cytosine. Just like there's autocorrect or word check on your word processor, we have really important proofreading enzymes that exist to make sure that we don't have base pair mismatches, that when we're creating this structure, everything goes correctly, we don't miss a spot. Because if we do, now we have a bad piece of DNA, which when it replicates is going to look even worse. And that's how we start to have mutation of things. So it's important that adenine, thymine, guanine, cytosine, these nice anti-parallel dual ladders with rungs, twisting in a very specific way, which I'm not going to get into the super hard science about. But yeah, feel free to Google anti-parallel, feel free to Google base pairs, and feel free to Google exercise and telomere length, because that's really cool. All right, folks, that was your first little lecture. I'll be at you with another one, and I will see you in class. Thank you so much for listening.